So, as you all are aware of about this Science Dialogue series, this is free webinar series, especially in biosciences stream. Purpose is to disseminate knowledge so that maximum people will get benefit, and that temperament will also generate scientific temperament, and people, those who are not privileged to have good quality of teacher, researchers, they will also get connection, networking, and knowledge from the top teachers from top institutions, reputed institutions. And sometimes some teachers, they may not be in reputed institute, but their credentials are very good. So generally we select the teacher who has confidence to teach online, and if they have some credential, better credential, confidence, because taking online lecture is not easy. Because in classroom you can handle your student and they are bound to listen you, but here if you are confident on your topic, then only you can take this risk to deliver lecture. So we generally request several people. Among those people, some people accept my invitation. Sometimes they are busy, or sometimes they are not confident, although they have hundreds of paper, but sometimes they are not confident to take online. So in this series, I welcome Dr. Sirisha Madam in the Science Dialogue series as a resource person, and we are grateful that you have accepted our invitation to deliver online lecture. For those who are new, those who have not seen the flyer, so Sirisha Madam is working as associate professor in Loyola Institute, that is autonomous institute affiliated to Osmania University. Madam has spent, she has more than decades experience of teaching and almost 1 decade of research experience. She has also published more than 10 papers. Voice is low. My voice is low. I don't think voice is low.
Little it is looking like that, sir.
Hello. Wait. My voice is low now. Okay. So Madam has also handled more than 100 students as a guide, their guide, MSc guide. So with this brief introduction, more introduction, Madam will give during lecture, because I don't give more introduction to a speaker, because I think their lecture will all will give their introduction, better introduction. I don't want to hype, and I don't want to diminish. That's why lecture will give that more introduction. But for attendees, I request all the attendees to attend complete lecture to know about the topic. And we also give e-certificate if you will attend complete lecture and fill that Google form. Generally we share in chat box. If you're not filling, then don't ask me e-certificate after 1 day or 2 day. So you must attend complete lecture. We also count the time, whether you are attending or not, because free certificate distribution is not purpose is to increase overall knowledge pool. So I welcome Sirisha Madam, and now Madam, platform is yours.
Thank you so much, sir, for your nice introduction. And I'd like to start and giving a chance to me for taking this lecture, sir. And I'm happy to share my knowledge with the friends and as well as with the students. So I like to start with the today's section with a quote. So your mind is the garden, and your thoughts are like the seeds. So the harvest can either be flowers or the weeds. So you can think in that way, and you can utilize your knowledge for what purpose you are doing your research. Sir, is it visible, sir?
Yes, Madam. Yes, full slide. So make it.
Yes, sir.
Yes, and hide this line. Click here to hide.
Yes,
Stop
sir.
sharing. Yes, Madam. Yes, it's very good.
Thank you, sir. Today I'm going to give little information about molecular plant protection in rice against bacterial blight pathogen. And how many of you are aware of this bacterial blight pathogen? uh, Actually this is a bacterial disease, so you can see that here the healthy panicle with grains, and now you can able to see this is a bacterial blight pathogen. Nowadays genome editing tools also they are using this xanthomonas oryzae pathor oryzae, that pathogen which caused this bacterial blight disease. You can able to see that right side the total crop is infected or infested with this particular bacterial blight. So now you can see that clarity how the disease is spreading. And neighboring to that one, you can also see the green color plants also there, rice plants. So you can clearly see the zone of that infected one and the healthy one, which gives that some genes are affecting major role for decreasing the disease. So introduction. As you know that rice is the scientific name is Oryza sativa, is the primary food source for half of the world's population. And India is the world's leading rice producer, followed by China and Bangladesh. Are you aware of this information? When I'm doing my PhD, and we are followed by the China. The first place is for the China, and then we come across the second place. Now we surplused and we moved forward against this China, and now 2022 our particular statistical data 9.8 million hectares or more. And now present scenario 24-25 149 million hectares we are growing compared to that China. So we are leading in a better manner compared to the China. Rice is grown in both temperate and tropical regions, so it undergoes number of abiotic and biotic stresses. And the abiotic stresses are caused by the non-living things. Biotic stresses are caused by that living things like stem borer, leaf folder, bacterial blight, and sheath blight, etc., which causes lot of crop losses. And so many scientists, so many members are working for this plant to sustain in the particular field against so many odd things. So we are thinking that only human beings are facing the stress content, but plants also are facing the lot of stress condition. Among biotic stresses, one of the major biotic stress constraint is the bacterial blight disease caused by the Xanthomonas pythovarese. The yield loss range from 10 to 80%. You can see that one, it is a great loss if this is affected. And now we can able to see that biotic and abiotic stresses which are caused to that rice. Now virus which will come under the biotic stress, bacteria, fungi, insects, and nematodes. So virus means rice plant tungro virus is the one disease. Another one is the bacteria. Here different bacteria, sheath blight is there, bacterial blight is there, and we are having the leaf streak. Different bacteria are affecting that rice plant. And fungi, when we are entering into that, we are having that blast disease which is caused by the Magnoporthe or Magnoporthe oryzae people used to call. Then coming to the next one is that insect. We are having that brown plant hopper, green plant hopper, which are causing a lot of diseases. And then because they will act as a vectors for the diseases, nematodes, gall midge. Now root nodules. So in generally we are looking into the dicot plants, but here in the rice monocots also, this gall midge is the infestation because of the nematodes which will accumulate in the root nodules, root area. And then abiotic stresses, and as you know that one is salinity, drought, heat, and cold. Salinity means already you know about very clearly that India is a peninsula structure. You know very well about that. So nowadays people are growing this fish pond culture or something. They are making that ponds near to the seashores. The intrusion of the salinity is increasing day by day, and water pockets are disappearing. The freshwater pocket zones are. Then drought condition. So many areas are affected with the drought, without water, less water content. Mainly in India, we are growing the plants with the help of rain, monsoon time and rain-fed areas only. We are looking for that. nowadays drip irrigation, and we are looking that one lift irrigation, different methods we are incorporating to increase that yield. And now we are present in the first rank because of these different whatever the technologies the government is giving, and people are farmers are utilizing, and they are securing in that way. And then heat, because temperate, and you can see that tropical regions, Africa, South Africa and other areas, a lot of heat will be there. And compared to that, the areas which are now you can see that Uttaranchal, or maybe you can able to see the areas very cold regions, like Germany, or you can see 6 months, and 6 months of heat, 6 months of cold and cold nature. These are the abiotic stresses which are really affecting that yield of the plant. Now, previously, when we are discussing about that, the land and water uses, you can see the clear picture depiction. Here, you can see the left side, plenty of water, plenty of area is there, and yield is okay for sustainability. But now, at present, we decreased the resources, like the land is reduced, and then water resources are reduced. Now we are expecting that plant to become more and more to produce. So now we are making that plant to give double and triple fold. So you can able to see that the stress which is giving to the plant very high. Now that SNSX, and you can able to see 2022, the rice producing countries, India, we are leading ahead than the compared to the China, Bangladesh, Indonesia and Vietnam. So history, when we are talking about that, there is no chemical effective against this bacterial blight. Therefore, the only way to protect the crop from bacterial blight disease is the use of resistant varieties. How to make a resistant variety? First of all, that is very, very important. When we are going to identify the resistant genes from the wild type, the plants which are grown in wild areas, maybe forests, or maybe that some ponds, and there we can able to find out these wild races of the rice. So we are going to cross with that whatever the cultivable varieties, or maybe present what we are growing that varieties. So there is a chance of getting a lot of other characters than the resistance. So our local varieties are giving that a high yield and taste and everything is good. The grains quality and taste, and so many parameters, physiological parameters we are looking for, that they are good in local varieties. But we are lack of these resistant genes, that we are going to cross with these resistant varieties which are there in the wild. Then they are turning into resistant varieties by doing that back crossing, BC4F6 or BC6F6. If you are going to see that back crossing and forwarding the crosses, then we are going to make that resistant genes to be fixed into the population. We are going to get the good yield of that. So breeding rice cultivars for resistance to bacterial blight is a high priority research area in both national and international agriculture institutions. So now the scientists, what they are doing, they are releasing the new varieties to look into that one, how this plant is going to sustain for a little longer, and which can able to give the good yield to the farmer, because the farmer is the main person who is working in the root level. And whatever the testing of that, the scientists who whatever they are making the new varieties, the actual leaders are the farmers who are growing in the fields, and against those stresses, and getting the good yield. So many resistant cultivars, especially with major gene resistance, have been released and adapted. So over time, however, virulent strains of the pathogen have overcome resistant cultivars. So I think all of you might have seen that one Tom and Jerry show. Okay. And why ma'am is talking about this Tom and Jerry show? Always, who wins the Tom and Jerry show? Anyone is having any idea? Always Jerry wins because it is looking for the life. Now pathogen also always wins because it want to survive. It has to survive in the particular location for a longer period. So whatever the plant you are making resistant against, incorporating the lot of genes, and this pathogen is smart enough to attack that resistant varieties and making them susceptible, or making them disease one. This phenomenon is a consequence of considerable shift in predominant pathogen race in response to the deployment of resistant varieties. Now we can able to see a lot of new varieties are coming. Some are there for few years, some are there for the months, like that. They are not able to withstand with this condition, and then now we can able to see that the variety may not be there. Sometimes we are losing that varieties because of the diseases. The cultivation modern rice cultivars that are highly responsive to nitrogenous fertilizers favors disease development and thus causes greater yield loss. Now, people are what they are looking at. If any plant, if you are providing the NPK ratio and nitrogen fertilizers, they will think that one plant will grow very high yield we will get. Simultaneously, the pathogen will also make use of this NPK ratio and it will increase its number. So at present, 46 major bacterial blight resistant genes have been identified in rice. Out of the 46 resistant genes, so much of scope is there to identify that new genes, because we are having around 3,000 genes are there in that. But among that one, we are having the 46 only they identified up to now. In the 46 also again we are having the 17 are recessive, remaining 29 are the dominant varieties, dominant genes which are we found. So here we are just genetics about the pathogenicity in Jew. We are going to discuss about this. What exactly this Jew? Jew means Xanthomonas varaceae, patho or varaceae. In short term, they will call it as a Jew. So Jew is a gram-negative proteobacteria. The pathogen colonizes xylem vessels with the bacterial cells and extracellular polysaccharides by invading the host through natural openings in leaves, including wounds and hydrothodes. As you know that one, when rice plant, if you whether you touch it or not, it is like a not a soft leaf, it will be coarse will be there. When they are rubbing again as the wind currents, a lot of cuts will be there. Through these cuts and stomatal openings, hydrothodes, the pathogen will enter into that plant and it will invade and it will cause great loss, because it will stay in the water for a longer period and then it will attack the plant through these openings. Factors are also released by bacteria which bind and activate the transcription of genes that activate resistant genes known as resistant genes. So we are going to find out that on what are those resistant genes, how they are responsible, whether they are going to give that much of resistance against the pathogen, how long it is going to give, that people are going to discuss about that. And nowadays we are having this what exactly genome editing tools we come across by using these talents also. So they isolated from the xanthomonas only. Out of 46 bacterial blight resistance, 11 of these have been cloned and characterized. They are XA1, XA3, XA26, XA4, 5, 10, all the resistant genes and resistant genes. Now you can able to see that out of 46, only we can able to make the 11 of them are cloned up to now. So now this is the present data, the condition. Now you can able to see here the DNA fingerprinting of that. Fingerprinting, you can see that this is an insertion element or the transposable element which is present randomly in the pathogen population, and that is taken into consideration and they made it as a marker, and then we are going to use this one with the study of this pathogen. Each lane 1, 2, 3, 4, 5, 6 numbers are there. Each one is having the one different bacteria. Now you can able to see this is that trap nursery. Trap nursery means people will grow different varieties of plants in that karuna susceptible variety, means disease prone, very disease prone. And again we are having IR24 is the background of the near isogenic lines. Now International Rice Research Institute made a NILS, near isogenic lines, which are having the combination of single or multiple genes. Now we can able to see that how at a 60% level now all the pathogen we cannot screen in the field, it will be very difficult task. So we have to make them as a group. Now 60% similarity, 50% similarity, which are in the molecular level, we are going to make it as a groups. And some are like individual isolates. You can able to see that here 16 number is there. Now this is a single isolate which is not coming under any group. Now we can able to see here also that this is the candidate isolates we will call it as, which are not coming under any group. Now we can see here this is the individual isolate and this is the individual isolate, we'll call it as a candidate. They are virulent strains which are not coming under any group. Now you can able to see this is a big population, And this big population, we can able to take one or two strains, not required more strain to screen in the field level. So now you can see here, and again that individual isolate, candidate isolate, we will call it as. So these candidate isolates are giving a crucial role, which is causing major disease in the plant rice crop. And this is another one, because I like to show the difference between that. Previously we used this Karuna and IR24, which are the susceptible varieties, how the molecular data you can able to see here. So which is giving the fingerprinting pattern of that. And next to, when you will see, this is the disease, you can see the culling of the leaf, and then water soaked regions. When it is turned into the curly way, no photosynthesis, and you won't get any particular grains in a proper manner. And here another picture which is depicting that hybrid rice. You can see that we will think that a hybrid rice are high yield and other. Even those also get affected by this bacterial pathogen. And now we can see that one, how this pathogen is. You can see that a beautiful fingerprinting of that one. It started with that one 100 base pairs to that one having the 14,000 base pairs we can able to see in a picture. And 60% similarity, again we are making into groups and we are taking isolates, and we are crossing to that means pathogenicity we are going to test in the field level, the plants which are having the resistant genes. Just as mentioned before, that one near isogenic lines released by the ERI, International Rice Research Institute, which is located in Philippines, Las Banos, we will call it as. And here, this is the background is the IR24. This is a susceptible variety. And now IRBB1 means one gene is there. That gene name is the XA1. And then IRBB3, XA3 is there. And then XA4, XA5, like that. We can able to see that different genes are located in single and multiple combinations. When we are crossing, means when we are doing a pathogenicity test, when we are cutting, dipping into that particular bacterial lysate and cutting this healthy plant, and within 15 days of time you can able to see that clear symptoms pattern of that. Now, this is a pathotyping test. You can see that 21 old plants were clip inoculated with the bacterial strain following clip inoculation method. 15 days after inoculation, the lesion length of formed due to inoculation was measured with a ruler. With a scale, we are going to measure that. Here, it is not like making any tantra mantra, and everything is that with a scale. Science is having the accuracy. We are going to test in that. Reaction showing lesion length less than 5 centimeters is the resistant one. You can see very clear of the first picture having the only dotted region is there. And then next coming to that one is a little more pathogen intrusion is there. When we are cutting with the pathogen content, then the disease, you can see that moderate susceptible. You can able to see that how the disease is spreading. Now you can able to see that the different plants are a different way of so which is showcasing that the genes are responsible for giving the resistance to that one. Now at that right side you can see that on total this leaf is affected with this particular bacterial blight. And when you are looking at the left side of this one, it is just only the dot, because it is less than 1 centimeter. It is like that. It shows that here the genes are playing major role to fight against the particular disease. And here also it is working nicely, but little more is required. More genes deployment is required, or pyramiding is required, so that we can able to make this plant sustainable in the particular area, diseased area. Now you can able to see the differences of that one, which is giving the clarity to you that how these genes are working against that particular. Now, what I mentioned previously, that hybrid rice variety, we will think that when they are high yielding and other. So now the how the diseases genes are working. IRBB3 means XA3 gene is there. IRBB4 means 4 gene is there, 5 gene is there. Now you can able to see that how that disease 12.5, 12.1, less than 5 centimeters means we can think it is a resistant one. Greater than 5 centimeters means we can think they are the susceptible one. Now we can able to see here only IRBB 10 and IRBB 21 is showcasing that resistant reaction. Remaining all are greater than the 5. Now like that you can able to see that one each variety, some are inbred lines are there, CMS lines are there, restorer lines are there, hybrid lines are there, all of them how the reaction rate will be, you can able to see the how the gene is working on that line. So that in general what people used to say that if foreign countries or maybe USA people, UK people or Germany people are doing any research, we like to implement in our area. But we have to look into that one how our pathogen population is spreading in our area, and which gene is responsible for that one. Once we will come to know, then we can able to club those resistant genes and we can make a new variety. Otherwise we are going to lose our local high yielding varieties instead of focusing on what others are doing. And first we have to study about that how pathogen is working. Now XF5 gene, this is a recessive gene which is in a double allele, it is working very efficiently. Now I like to show us this one. Now this is the marker, low range marker we are going to use, and then a resistant band pattern. Now we can able to see that one here the band pattern at 430 base pairs, and then susceptible one you can able to see here the band pattern at 400, like that it is. Okay, and then you can see that 2, 3, 4, 5 heterozygous condition. So it is a SSR marker, simple sequence repeats, and generally this codominant nature may not be there for the each marker. Then we have to make them converting that dominant nature marker into the codominant nature marker. So we are going to digest with the enzymes. So now this is a XFI gene. When you are going to do the PCR, you will get only one band. So dominant gene means a dominant trait only will appear, and recessive trait will not be seen in that particular picture. So what we have to do again, we are going to digest with that enzymes, restriction enzymes, that will give the clear pattern, and which is that a susceptible one and which are having that one heterozygous condition. Now can able to see that how the heterozygosity is looking into that, which is helpful for the researcher breeder to identify that which plant is having the heterozygous condition, which is having the dominant resistant condition and a susceptible condition. So in general, when you are looking at the dominant condition of the plant and then heterozygote, both looks similar in phenotypically, because they look like 1:1 condition. When you will see that again it will segregate into 1:2:1. Then one is the dominant condition, one is the recessive one, but the two condition which is representing the heterozygous condition, which we cannot see with our naked eye, and it will be very difficult to study inside what exactly that how the pattern is DNA, how this condition of the polymorphism is there, we cannot see. So unless until when we will work in the wet lab, then only we will come to know how this pattern, and which will give the clarity. Again, next generation it will segregate into 1:2:1 ratio. Then coming to the next gene, which is the XA13 gene, which is also a recessive gene, which is also doing nowadays in India, we are working mainly on the XA5, 13 and XA21. XA5 and 13 are the recessive genes. XA21 is the dominant gene. Now you can able to see here, this is a susceptible one is at the 700 base pairs, and then resistant band pair is that one 400 base pairs. Now wherever you will see that one, and then you can see that one 3, 4, 7, and 10, 11, and then you can able to see the 15. These are the heterozygous condition. Even we can able to see that this is also previously it is a dominant marker, and then after digestion with the DRA gene. So previously it was the DRA gene we used, and here we are using the HINF gene 1. So HINF 1 is looking for cutting of the particular band and the polymorphism, turning into the dominant gene, turning into that codominant nature, which we can able to see the clear differentiation, having that one susceptible nature, resistant nature, as well as the heterozygous nature. And then coming to the next one is the XA21. And in India, because when I'm doing my PhD, I worked on these lines. XA21 is a very predominant, dominant gene, which is giving lot of resistance. Most of the farmers, they grow no, only XA21 only. Their demand is that one, we require the plant with the XA21 gene. Now you can able to see that the susceptible one band is there at 800 base pairs, and resistant band is that 1000 base pairs. Previously, it was around say, 10 centimorgans away. The marker is identified, PTA marker 248 something. Then later, they found that when crossing over occurs, the marker will be situated in other place. So they made the gene itself is the marker. Now, whatever the polymorphism you're looking, if this highlighted the fragment means it is the gene is highlighted, which is there in the recessive condition, which is there in the resistant condition. Now you can able to see here in the third, 4, 5, 6, 7, 8 conditions, we can see that a heterozygous condition. So now we can able to see that clarity of the things, and in bacterial leaf blight trap nursery, now CRRI turn into a change it to the NRRI. Now you can able to see that IRBB 21, IRBB 21 having the resistant gene of XA21 gene. Now he's showcasing that the lines you can see that one with the green, and having that susceptible variety Karuna. Karuna is a really high yielding variety. The taste of the grains and the long grains and everything is beautiful, but lack of disease resistance. So now we are going to incorporate that resistant genes into our local varieties. It turned into the resistant cultivar, and the farmers are really interested for growing their local varieties. Now you can see the clear differentiation of that one diseased one and the healthy one, which is having the resistant gene. And then this is the once upon a time the scenario. Now the scenario been modified, changed. Then now you can able to see this paper I published a long back. Susceptibility of IRBB 21 carrying the resistant gene XA21 to bacterial blight. What happened here? Now you can able to see that one. Just know what I told, that one, this is dominant gene. It is giving a lot of resistance. Now the pathogen become over smart, and it is attacking that particular plant and making it to the susceptible. Now you can able to see that which the plant we thought of that resistant having the 6.8, 11.2, 7.2, and 8.5, 6.4, 10. So greater than 5 it is there. So you can able to see that one. The genes are the genes condition in resistant condition. Resistant genes are present in that, but total failure because the pathogen is a turning to that virulent strain. Virulent strain means which is causing more disease. So already I told you that previously, whatever the varieties you are growing, if you are growing one type of varieties, the disease will be the pathogen attack will be very high. If you are growing the different varieties, it takes little time to spread the disease in the particular area. Now, the whatever the dominant gene, IRBB 21 having the XA21 is a failed because people, the farmers, when they found that the symptoms, they applied lot of NPK ratio. They thought that one, the giving that nitrogen fertilizer will improve the growth of a plant. But all of a sudden, what happened? The pathogen population increased a little more than that because it is utilizing this NPK ratio. NPK is helpful not only to the plants, even pathogen multiplication also it is required. Now, when I'm doing these are my lines, what I have done in my PhD. Now you can able to see that these are the IR64 line, high line variety. And now you can able to see here, this is the already clip inoculated. When we inoculated the local varieties, adjacent to that one, having a lot of infection, but our plants are survived well. And how long I cannot give you the guarantee, but we pyramided XA5, 13, and 21 genes in our area because the genes are working very efficiently. And then you can see that this is the disease condition. We like to turn this disease condition to this healthy rice crop condition. So thank you so much. And any doubts or anything is there, let me know that. And we like to discuss more about this.
थैंक
And
यू
we,
मैडम, थैंक यू। मेरी आवाज आ रही है?
हाँ sir.
यस, थैंक यू वेरी
So
मच। नाउ आई रिक्वेस्ट attendees to ask question